developmental origins of chronic disease. And her research interests are in the early infant nutrition and particularly microbiome influences on later chronic disease, particularly allergy and asthma. She's very involved with the local breastfeeding community of Canada, and especially with Ishmael, and a dear friend. And I want to publicly thank you for organizing such an amazing event. I've already learned so much and met so many people. I'm really excited about the outcome from this event. So thanks to both of you, but please welcome Meghan.
Thank you very much, Bridget, and like Nathan, I invited myself, so there's no one else to thank, but to thank all of you for coming. So I want to start with a little bit about kind of how I got here meeting milk and lactation science and research. So I know that for a lot of people in the room, this started with a personal experience of breastfeeding. So being in a different field, having a baby, breastfeeding, and realizing what an interesting area it might be. So I don't have that personal experience. But I have personal experience in another way, and it started with data. So I was studying the infant microbiome, and this is one of my first papers on that topic. And so I was studying the microbiome, and that was really my interest. But it was just so unmistakable and interesting that of all the factors we were considering in terms of shaping the microbiome and believing that that's important, breastfeeding was like far and away so much more important than anything else. So we were thinking about antibiotics, which of course are made to kill bacteria, method of delivery, and these are important things too, but really breastfeeding just surpassed them all. So when I started my own career or research lab, once I got my faculty position, I really decided to focus on that. And then I have experience with other researchers. So I was really new to the breastfeeding field because I came at it sideways from microbiome. But in meeting different researchers, initially at microbiome conferences, so Lars is one of those examples, kind of meeting these other researchers in the lactation field, and many of you are people I've met at other conferences, and just recognizing how nice of a community it is and how interesting all the research is, really has motivated me to dig deeper into the field and stick around. I've had some experience with media, so when this paper came out, it was publicized, and I remember talking to the press officer at our university who wrote the press release, and we explained in lay language to her what our findings were, and she said, okay, I think I got it. So babies who are vaginally delivered have a different microbiome, babies who are breastfed have a different microbiome. Okay, I got it. Is there anything you think people might get upset about or take negatively? And at the time, I thought, well, of course not. This is just cool science. Everyone will be happy to hear this. No controversy at all. And clearly, I didn't have personal experience, and I had little experience in the field, and the onslaught after that was something I will never forget. I try not to read comments anymore on articles online, but why are you blaming us? It wasn't our choice to have a C-section or to formula feed, and get off your soapbox, and all these kinds of comments. So it's been an interesting kind of realization and journey with that type of attention. And then experience with moms. So over the sort of 5 years I've been in this field, many of my own friends have had their first babies. The conversations with them has been interesting about birth and breastfeeding and its importance and why we're studying it, and taking more into account their feelings and perspectives about this research. And then care providers too. So having done a lot of research on the microbiome and the components of milk, and from a purely scientific view, thinking how interesting it is, to realizing, like Kathy just showed, that there's this whole pyramid, and at the top is the milk composition where I focus a lot of energy, but trickling down from that are these other really important layers, and part of that is the care providers. So I joined the Baby-Friendly Hospital Implementation Committee. So our hospital here is not baby-friendly, but they're working towards it, and I thought maybe I could help. So I sat in on some of their committee meetings, and for me, and this is probably not surprising at all to some of you who are care providers, but it was shocking to me to hear that we can't implement breastfeeding within 1 hour because the nurses have 10 other jobs to do, or the formula bottles are this way, or we have funding from this contract, and it was just nothing at all to do with the scientific numbers. So I've been spending all this time crunching the numbers in my computer, believing that once I found the association or the answer, that everyone would just implement it. It would be simple. And of course it's not, but I didn't recognize that necessarily when I started out. So the main research questions that I'm working with now, many of them are done within the context of this child study, so it's a pregnancy cohort. This is where I did my initial research on the microbiome. so it's very close to my heart. It's a national study. We have 3,500 families participating across Canada, including 1,000 from here in Manitoba. It's a very broad study looking at developmental origins of disease. There are many facets to it, many researchers involved. Fortunately, they collected stool without really knowing that it would become so important, but that's where the microbiome research has really exploded from. And then, like I say, when I started my lab, I realized we had also collected breast milk, and while a whole bunch of people were fighting over the poop because the microbiome was so important, nobody was doing anything with the milk. And I thought, this is a missed opportunity, I will start studying it. And so that's how I got into first the microbiome and then this angle of nutrition and diet. So personally in my research group and with my team, some of the students are here, we're looking at questions around epidemiology or associations of breast milk feeding, and that's put in there purposely because we've looked at things like pumped milk versus direct breastfeeding or nursing, and associations with all of these different chronic diseases that we're monitoring in the study. But then layering underneath that the milk composition. So we have those samples, we can look at the components. And then layering beneath or on top of that all the maternal factors that might influence milk composition, might influence a mother's ability or decision to breastfeed, and might separately through completely unrelated mechanisms influence child health. And then we can start to look at mechanisms. So if there are certain components that are important in the milk, we have the stool, we have blood, we have immune profiles to be able to look for those connecting mechanisms. I do want to really use this information for interventions at the end of the day. The knowledge is important and interesting, but I think we need to mobilize it beyond that and to say how can we either optimize milk if that's possible, or address those underlying factors, or support breastfeeding in other ways. I do a lot of this in the child study cohort, but I've got a mouse on there because I do work with scientists who do animal models, and I've got, hopefully now you all can recognize the province of Manitoba because you're here, but I work with Nathan and others with administrative data at the population level. And so this is not to read, but this is some of the questions we've asked in the child study. So on the one hand, this is very detailed information about feeding, what kind of bottles are used, if there's pumping, what kind of formula, if there's formula. On the other hand, though, it's not as detailed as maybe I would like, right? So we didn't collect the exact proportion of formula versus pumped milk and so on. But a decent number of questions, and some of the details that we're digging into is separating, for example, nursing at the breast from breast milk in the bottle, and starting to show that those aren't equivalent in terms of certain health outcomes. Then with the composition, this is, I think, a nice visual of the differences between milk and formula, and some of the components that we've measured. I have a growing wish list of the other things I'd like to measure, so we've done, and all of this is in collaboration with other researchers who I've met through ISHRAL and other networks. So oligosaccharides with Lars, bacteria, and just recently fungi with my student Sharon and a collaborator here in Manitoba who comes from the dairy milk science world. So that kind of cross-collaboration has been important too. Fatty acids with Catherine Field in Alberta, as well as endocrine hormones. Peptides and cytokines are two new items on the list that I've just been funded to do with collaborators in the Netherlands and here in Canada. So really excited about those. Thinking about the moving beyond the mechanisms into these interventions, I agree with Aloka who talked about the education, and Amy talked about it too, this idea that if we think more broadly, we need for a society to be supporting breastfeeding, and for society to be supporting breastfeeding, we need to first know that it's normal and it's beneficial and it's important, and that's not just women, that's everyone, and I think probably needs to start really young. So I have a little side project unfunded to look at education of around breastfeeding in schools. So what I'm really excited about, everything I just said, but also a new thing which Nathan mentioned, is our Manitoba Interdisciplinary Lactation Center, or MILC. It's an honor and really exciting to have Tina Chambers here because we are modeling our biorepository after what she's accomplished in San Diego, which I think is fantastic. So this idea of building up a biorepository of milk samples that are available for research, and that it will be linked to the database that Nathan described, where he's collecting this information about infant feeding. So really powerful, I think, when we bring it all together. So that's kind of launching this year. Another thing I'm excited about, which I haven't talked to too many people about, is something still in the planning phase, but we're really excited about. This would be an intervention study. I do work on allergies, and we have some observational data to suggest that maternal consumption of peanuts is important for preventing peanut allergy. And so these are observational data, but we really need a trial to test that hypothesis. So we've had some interest from the NIH, which is encouraging. So we've been down once to pitch our study, and we're kind of refining it. So I'm really excited about this. Next big breakthroughs. So a couple that I think about. One is really the bigger picture of milk, not just one component at a time, but the framework. So this is one framework developed by Sharon and Shelley. I promise it's almost submitted like it said there for the special issue. But this idea that we have to think about the environment, milk itself, and health, and all of these things and how they interact together. So Sharon's been learning structural equation modeling and ways of looking at these in a more complex picture. I put out there too multi-omics, so this idea that we have the microbiome, but also the oligosaccharides and fatty acids, and how do we think about these all together? I really think too on the biorepository angle, so we have one great biorepository in San Diego, we're starting up one here in Winnipeg. If we can get really a global network of these biorepositories, it would really facilitate a lot of important research around what milk looks like in different parts of the world and what's important to be considering in those different settings. And I think we can be creative and innovative about the way that we collect this milk and link it to other database sources. I'm also really excited about the milk GWAS that we're doing. So we have genetic data in the child study cohort and looking at those genetics and how they influence milk composition. Personalization of donor milk. So this is a project that I'm working on with Marilee to develop. We're conceptualizing a trial that would start, taking small steps towards this idea of not just pooling donor milk and giving it to all the babies that need it, but being more intentional about which donors are matched with which recipient babies. That's a multi-omics diagram. So barriers, I think, for me, being transdisciplinary is, I love that about my research, but it's also my biggest challenge. One is prioritizing projects. So Kathy was talking about having one exciting project, but it taking a long time to come to fruition and having many things that you're excited about. So I do have many things that I'm excited about, and my challenge now is prioritizing them. So something might be exciting, but then when I took a step back to say, but the time and energy I'll invest in this, what impact is it going to have? And then starting to recognize that I have only so many hours in the day, and what should I really focus on? So having discussions like this today with all of you helps me prioritize that, so thank you. Straddling the basic science and the population health. I don't think I'm the only one doing that, but I think in many cases people are more on one side than the other. And I like being in the middle, but that also makes me feel that no matter what I do, someone somewhere is always upset, and maybe we can never get around that, but it's an interesting place to be, straddling those two places. I think it's important. Knowledge translation to diverse stakeholders. So there are so many stakeholders on both sides of that, and understanding the best way to get the knowledge out is a challenge. And then a more practical one is just finding good trainees. I have so many projects and ideas, but finding good people to work on them. So I think this is where the TAP program maybe comes in, and maybe we can, as a network, kind of share around our training. So for the field, they're similar. So I put trainees up there again, and we're asked to think about solutions. So I think programs like TAP are one way of getting at that. The prioritizing projects issue is for me, but I think as a field too, what do we want to prioritize? So maybe systematic reviews are a good way to set the state of the knowledge and what needs to be asked about. And then I think networking is so important, and we're doing some of that here today. Interdisciplinary research. These are things that we'll discuss in more detail tomorrow, but the conflicts or confrontations, or maybe those aren't quite the right words, but between the camps of mothers, Amy talked about this, the mothers who couldn't breastfeed, and instead of fighting amongst each other, kind of going towards a common goal, and then sort of conflicts and confrontations between the basic science community and sometimes the population health community. I think those can work together well, but so often they come to conflicts, and I think it would be good to make more progress as a team. So I think I'm almost at that on time. I want to acknowledge everyone. I have so many collaborators. Everything I do is in collaboration with others. I have a great team. Some of them are here today. I hope you've had a chance to chat with them. And for the child study in particular, all the families who participate. So thank you.
So I have the pleasure now of introducing Dr. McGuire. I'm going to share a quick story last night. She's like, oh, it's so nice to meet you. And I fangirled Shelly, so I was like, oh, I know who you are. Okay. So I've been following Shelly's work for years. She's a professor of nutrition phenol in human milk and how that impacts our understanding of what we still need to know and infant health. And she's also the PI of the INSPIRE study, which is really on the cutting edge of moving the field forward. And so it is my pleasure to welcome you. Here is your presentation. Your slide of answer if you like it. Thank you.
Well, I too am very grateful for being invited, so thank you so much. And I know that we're like 2 talks away from getting to go outside, so I'll try to get through this in no time flat. I have embedded, hopefully, the answers to the questions within my talk. And really what I would like to do today is sort of introduce Team McGuire, because I don't know most people here, and I thought this would just be a good way to sort of let you know what we do, what we're working on, what our challenges are, etc. So that works. These are my conflicts of interest and gratitude disclosure for the funders that we currently have. And we'll start off by just introducing the McGuire team. So this is my husband, Mark McGuire. We've been married for quite some time, and as you can see, we actually, we toasted with milk. Like, that's a real picture. Mark is a dairy scientist, also got his PhD in nutrition and lactation biology at Cornell. So we've been together a very, very long time and have been studying milk. And so he's my #1 collaborator as well. So this was just us a few weeks ago, a month or so ago, at the USDA, actually talking about science communications and how scientists need to get more involved in that. So we've been at this game for quite some time. We run a relatively small collaborative research group at the University of Idaho with another PI, Janet Williams, probably many of you know her as well. So we do team science, we collaborate, and we do interdisciplinary cross-species work, mostly related to lactation and nutrition. So just to give you a sense of what we have been doing and what we're doing now, and you can see that I think our past experiences doing research have informed how we do our research today. But for a long time, we studied regulation of milk lipids, and mostly we were interested in conjugated linoleic acid, which is a really interesting fatty acid, and other trans fatty acids. We studied milk composition in cows and women. We tended to use we did very controlled dietary intervention studies. That's sort of our forte. Small scale Latin square crossover sort of design. So very, very controlled studies. We have quite a bit of experience doing stable isotope work, and we did all of our work in this beautiful little area of the world, Moscow, Idaho, and Pullman, Washington. So we spent over a decade doing work like that together. And about 10 years ago, a little less than 10 years ago, that really all changed because we were doing an intervention study looking at the effect of CLA supplementation on mammary inflammation. And during that study, we were collecting milk samples. We were looking at the milk samples under the microscope to count cells and try to understand the immune cells in milk. And we also sent the milk samples to the vet school to have the milk analyzed for bacteria because we were interested in mastitis and inflammation. And what was interesting is that the lab came back and said, all your milk is sterile. And we said, well, but it's not, because when we look at it under the microscope, we see bacteria. So we're not microbiologists, this is not what we do, but this was at the beginning of the Human Microbiome Project. And so we said, oh, well, maybe there are bacteria in there that won't grow using culture-dependent methods, and that is true. So we sent the milk to get sequenced, and this is just a stack bar chart picture of the data from that study, and I won't go through the details, but this ended up being the first study published using next-gen sequencing to look at the milk microbiome completely by accident, right? So opened a brand new field of research for us. We thought this was fascinating because we all thought milk was sterile at that point. And I guess I'm making this point here because it really changed what we do in the lab, how we do it, and where we do it. And so for those of you who are sort of at a new phase in your career, don't assume that what you're doing now is going to be what you're doing in 10 or 20 years. It very well might change. So I was asked to speak a little bit about the INSPIRE project today because this is an interdisciplinary international study The Inspire project was born from that first paper where we recognized that there is this rich microbiome in milk. And what we did in the study is, well, we back up a little bit. We published that study within 2 years, within a year. There were a couple other papers looking at the milk microbiome from other places in the world, and we got very different results. And so this was sort of the beginning of the field, and we decided, hey, let's just all get together and do the study right. Let's see if we can design a study where we're looking at milk microbiome all over the world using the same methods of collection, analysis, etc. So it was really a labor of love by a small cohort of what we call friends and family that were able to collect milk samples exactly the same way, and then we analyzed them the same way. So in this study, we tackled the question of what's normal in terms of milk microbiome. Lars was a major person, I don't know where Lars is now, but he was our HMO guy. We were also interested in infant fecal microbiome and some other factors in milk. And what was really important to this study is we had anthropologists on the team, and we have collected an amazing set of metadata that we've hardly tapped. So it was a combination of doing basic science and also looking at anthropology. So the basic study design, which you'll see is completely different from the sort of research that we had done in the past. This is a cross-sectional, cross-cultural epidemiologic study, no interventions. Oh, I like interventions. You can do good statistics, easy statistics. The populations that we looked at, again, these were based just on who we knew around the world and who we could trust to collect a milk sample. So we had 2 groups in Ethiopia, 2 in the Gambia, 1 in Ghana, Kenya, Peru, Spain, Sweden, U.S. California cohort, which Lars oversaw, and then our cohort in Washington and Idaho. And there were about 40 women in each group. So we ended up with 412 relatively healthy moms and babies around the world. We have no idea if time postpartum affects milk microbiome, so we standardized and collected the samples between 1 and 3 months postpartum. And this just, so this harkens back to our controlled study sort of thing, right? We controlled everything, down to what diaper, people put on their babies and how they cleaned the breast, cleaned the baby's bottom, etc. We sent all these kits around the world, and then the milk and fecal samples were sent back to us, and we analyzed them, sent to California for HMO, etc. So what we were able to do with this study is that if we found differences, we were pretty sure they were real, which sounds like a no-brainer, but it's easier said than done. So I'm not going to go through the Inspire team, but obviously this is a big group effort around the world. And what's I think important is the interdisciplinary nature of the study, which I think is something that we absolutely have to do as we move forward as much as possible. So I'm a nutritionist, we have lactation physiologists, immunologists, anthropologists, microbiologists, epidemiologists, etc. on this team, and this is growing as we continue to find new things to do with our milk samples. So I think this is critical, talking about the future of human milk and lactation research. So just really quickly, a few selected findings. I'm not going to go into a lot of detail. The first paper that came out, we were looking at immune factors, and the cohorts are on the Ethiopia down to the United States in the bottom. And I'm not going to go through the details, but we found massive differences and surprising differences. So for example, the IgA levels in the United States and in Sweden were much higher than the other cohorts. We followed this up with some additional studies. This is actually, without going through the details, what we were interested in was, well, okay, we have differences in total IgA and IgG, but what are those antibodies actually targeting? And we teamed up with a group in California who's able to look at specific antigens and then look at immunospecificity in those milk samples. And long story short, if you do that sort of analysis, you find really nice clustering based on regions of the world. So not only were the amounts of antibodies different, but what they were targeting was also different. and we got the kind of clustering by region that you would expect. So not to go into detail, but the bottom line is there are differences around the world. This was the second paper that we published. Again, Lars did the analyses and helped us understand them. Again, these are stack bar charts with the different cohorts. Details aren't important. The bottom line is lots of variability around the world, both in total amounts of HMO and also in the profiles. Same thing with milk microbiome. All you need to look at here are the differences in color in the bars, the different color patterns. You can see some similarities. You can see some big differences. So this was our main focus for the study. The question was, does milk microbiome, is it consistent around the world? And the answer is it definitely is not. And here are the infant fecal microbiomes, actually more consistent around the world, but again, we see differences, and we're trying to now figure out what's driving those differences, or at least what is related to the variability. So conclusions so far, guess what? Milk composition is variable. And I still say, that is not rocket science. We knew that. But seriously, unless you do a study like this where you try to control for everything you can control for, you can still come up with reasons why you're getting differences that have nothing to do with reality. So simple conclusion. What's driving this variation? This is what we're trying to look at now. We have a lot of information on diet, environmental exposures, even childcare behaviors and choices. My colleague, Courtney Meehan, is looking at, does it matter how many people hold the baby or how often the baby is put down, whether the baby sleeps with the mom, etc., whether there are chickens in the house, that sort of thing. And like Meghan, we are so excited because we're doing Do Us. It's just like a whole new world, right? Really cool. And then I think the real question is, so what, right? So as we were in our little group over here this morning, we're talking about what needs to be done, how does it need to be done, but in the end, what we really want to do is to be able to relate variation in milk composition to variation in infant and maternal health. That's where we're going, and unfortunately, that's where the funding agencies want us to go to quickly before we do the basic research. So challenges and barriers, I think, are completely related to that. And you've heard this before today. We are frustrated and have been for years that seems like the funding agencies seem to think we already have the basic research down. And like we always tell our grad students, that's why we call it research. You have to keep doing it. You have to go back. You have to do it again. You have to do it better. You have to look at it another way. And we know that, and how we're going to get the funding agencies to recognize this and let us do the basic research that we need to do, maybe for new milk components, it's tricky, and I think it's something maybe we can do better as a team. So this basic assumption that the basic science has been completed is so far from the truth. And then I think another barrier is that so many people want to put that proverbial cart before the horse. We want to relate something to infant health, right? But we still don't know how to collect the sample right. We don't know how to analyze it right. There's so many things that we still need to learn how to do. It's just, again, get the basic stuff done before we move to the more applied. And then something that some of us are going to be talking about tomorrow, I think another challenge that we have here is that the public climate has unfortunately turned to sort of being distrustful in terms of scientists. And so on top of everything else we have to deal with right now, we have to worry about things like who's funding our graduate students and where we're getting research funding from, because part of a large proportion of the public would like to discredit us if we've done anything that they seem to think is inappropriate in this regard. So it's just another level of added challenge and barrier. So with that, I would like to again thank the organizers for inviting me. And I put this picture up here because I haven't talked about our leprosy work, which seems totally crazy that we're doing leprosy work, but you can ask me about that maybe on the walk or something. So thank you.
Lars' work has been seminal to our understanding not only of the benefits and the presence and the characterization of human milk oligosaccharides in human milk, but also to our understanding of the etiology of necrotizing enterocolitis and other infant outcomes that are directly related to human milk oligosaccharides. And he has secured over $10 million in funding from various agencies. So I think we have a lot to learn from you when we talk about our challenges, including myself, in seeking funding. So I'm excited to hear what you have to say.
All right, thank you very much. All right, thanks for having me and thanks for organizing this workshop. This is amazing. I think it's a great idea to put this together. It's been very nice, very smooth, too smooth almost. I haven't gotten any hate emails before. No threats on social media. Something's wrong with this one. Yeah, I'll get to that.
Tell us to put our social media away.
All right, so I religiously followed those 5 questions that you gave me. I'm German, so I have to kind of follow the rules. So I go really through this, what is my research questions that I'm trying to tackle with my group. And as you said, we're working on human milk oligosaccharides, or short HMOs. And just a couple slides to introduce that. These are the main macromolecules in human milk: protein, fat, and lactose is the milk sugar. And then there's oligosaccharides, 5 to 15 grams, which exceeds the amount of total protein. So comparing that to bovine milk, hardly any oligosaccharides in there. And since bovine milk is the basis for most infant formula, we have hardly any of these human milk oligosaccharides in infant formula. So there's a real, just from the composition, a huge difference between breast milk and formula. So when we say human milk oligosaccharides, it's not just one structure. It's like saying human milk proteins, and that's all the same. There's different proteins. So we have different oligosaccharides as well. In fact, we have about 150 to 200 different oligosaccharides. And the important part is that every mom makes a different profile of these oligosaccharides. So talking about what's normal, normal is this beautiful variation. It's all over the place. And in fact, there is very strong differences. If you see a relative abundance, and if you see this here in this PCA plot, we move this around. So here, the closer the dots together, the more similar the oligosaccharide composition. The further apart, the more dissimilar. You can see there's huge differences. There's these 2 clusters, for example. So does that mean that one cluster is more normal than the other? No, that's what we find in nature. There is about 25% of women that cluster in that one area. When it comes to oligosaccharides, and the other 75% is on the other side. Is one mom better than the other? I don't think so. So the main interests really are what drives that variation. What are the maternal drivers, genetics and environment, to contribute to that variation? Does it change over the course of lactation between the same mom, within the same mom? And then what does it really mean? What do these oligosaccharides do in terms of infant outcome and outcome that is not just related to the infant, that is either the infant was breastfed or not, and what happens to that person when it becomes an adolescent or adult? And really, when we study this, do we study individual oligosaccharides? I told you there's 150 different ones. Do we just focus on 1 or 2? Or do we have to take them as a combination of multiple different oligosaccharides and their relation to each other? And then there's not only oligosaccharides. Human milk is more than oligosaccharides. Of course, we like to think that's the most important component, but of course it's not. It acts in combination with everything else that is in human milk. So I think that is a major challenge. And we're really looking through this, the health implications of HMOs throughout the life cycle. When we think of breastfeeding, we think of the infant that is breastfed at that moment, so the immediate short-term effects. But we also know that there is the whole DOHAT concept, that there is long-term effects of what the infant is exposed to during the breastfeeding period. Then we also know that oligosaccharides are already in pregnant women's circulation. So we find oligosaccharides at the end of the first trimester in mom's circulation. So there might be effects of oligosaccharides on mom during breastfeeding and during pregnancy, and that might have some long-term effects as well. We've also recently shown that oligosaccharides are found in amniotic fluid. So it's not that the baby is exposed to oligosaccharides only during breastfeeding, it happens already in utero. So here we're not talking about breastfeeding or formula feeding. This baby doesn't have a choice, it gets exposed to the oligosaccharides, but to which ones? And what does that really mean? So is there any benefits already during the period in utero, and are there any long-term consequences of that? And And then we have these new synthetic oligosaccharides that are becoming available at scale and at cost, that we can take these oligosaccharides in real intervention studies now, in babies, but also in adults. So we use that as therapeutics for diseases that are completely unrelated to breastfeeding, but where we can learn from the breastfeeding period and develop new therapeutics, for example, for the elderly with arthritis, atherosclerosis, you name it. All right, so what's the one thing related to this research that I'm super excited about? One? Only one? All right, so I can name a bunch. I start with how does this really. So I started working on oligosaccharides about 20 years ago. No one cared about oligosaccharides 20 years ago. Most people couldn't pronounce it. And there were maybe 3 groups in the world that really focused on doing oligosaccharide, and one of them was where I did my PhD. So really this whole hype on the microbiome, or the microbiome wave, really pushed the oligosaccharide research as well, because it's one of the contributors to shaping microbial communities early on in life. So this whole prebiotic notion of oligosaccharides really pushed the emphasis on oligosaccharides. So that's the reason why oligosaccharides now get funded apparently more than anything else. That's great. What also is exciting is the recent availability of individual oligosaccharides. So when I did my PhD, we calculated if we took an oligosaccharide and we wanted to take that for our studies, that was always a concern. My PI would always be like, oh my God, do we really have to order this oligosaccharide now? In milligram, really? Do we need a full milligram? Can't we do 100 micrograms? Now these oligosaccharides sit on the shelf in tonic form. Very low price. In fact, the point where they're now introduced in infant formula, which comes with all kinds of other complications. I'm getting into that in a bit. But really it allows us to do research using these oligosaccharides in animal models, for example. And then the other thing that I'm excited about, what really happened over the last few years, when I started my lab, about almost 10 years ago, we were able to analyze milk samples, 8 samples a week. So now imagine doing cohort studies with 1,200 samples, 600 samples. That grad student would just run out the door. It's like, I'm not analyzing this, takes me forever. So we've optimized this, that we are right now about 150 to 300 samples a week. So now we can really talk about analyzing large cohorts and investigating what maternal factors drive oligosaccharide composition. And how does that oligosaccharide composition now impact infant health? So that really helps us to generate new hypotheses. And then what I'm super excited about is collaborations inside the field, with some of you here in the room, but really also with people that have nothing to do with human milk, or had nothing to do with human milk research. The guy that studies arthritis, the guy that studies atherosclerosis, all of a sudden thinks about human milk. Like on our campus, for example, it's crazy. Everyone wants to do human milk research right now, because they really see the potential to apply that to their research as well. And I think that's great. All right, so what's the next breakthroughs? I think really better understanding how human milk oligosaccharides contribute in shaping microbial community and the effects that come out of that. I think that's going to be one of the next frontiers that we already kind of added. But also better understanding all the other microbiome independent mechanisms and effects. Not everything has to do with the microbiome, although it sounds like it right now. There was a world before the word microbiome existed, and I believe there's many, many other effects that are unrelated to the microbiome. Then advanced HMO synthesis and application especially in the adult space. I think that is one of the next breakthroughs. And then the link to other large data sets, either analytical data, so microbiome data, for example, of any assets we talked about earlier, and then all the clinical metadata that we have. And what is the biggest challenge? The same thing. Link to other large data sets. I think that's a huge challenge, and some of you brought this up before. How do we connect all these dots? We have all this massive data, how do we make sense of it? And I think that's going to be one of the major challenges, and at the same time opportunities for the future. What I am a bit concerned about is the incorrect data that's kind of muddying the field a little bit. It's the hot field right now, so everyone wants to jump on it. Unfortunately, it comes with, for example, people working with the oligosaccharides that are not the purest. I'll give you an example of that, with 2 examples. One is, if we want to study oligosaccharides, we want to study oligosaccharides and not lactose. So you see, lactose is bacteria will grow very different. You get very different results if you use different types of oligosaccharides. So I think that's very important to know what you're actually working with. Another example for a contamination is LPS. It's a lipopolysaccharide. Okay, oligosaccharides, polysaccharide, they're very similar in their structural composition. And LPS is everywhere. It's extremely resistant, very difficult to get rid of. It does accumulate during the HMO isolation and during production as well, because most of these oligosaccharides are made in microbes. So you have a lot of LPS in there, and that's very important. Here's an example from work that we've done on macrophages in the mouse, where you stimulate the macrophage with LPS to get the cytokine levels up. You see that. Okay. So that's the second part of LPS. And then we use the same oligosaccharide from 4 different vendors and threw that on the assay. And you say in 3 cases you get a pro-inflammatory effect of your oligosaccharide, and in 1 case you get an anti-inflammatory effect. It turns out those 3 were highly contaminated with LPS. So yes, they give you a nice pro-inflammatory effect, but that's the contamination. If you clean up these oligosaccharides and remove the LPS, they all give you a nice anti-inflammatory effect. And those studies are published, so you find out there's pro-inflammatory effects. And that's really muddying the field quite a bit. All right, conflict of interest is very important as well, of course, which brings me to the next point, the human experiment. So I mentioned that oligosaccharides are now available at scale to be added to infant formula. We're doing that right now. We're adding 1 specific oligosaccharide, 2-funcsyl-lactose, to infant formula. It's on the shelf. It says in big letters HMO, in small letters 2-funcsyl-lactose. And we have no idea what it does. We don't know what the short and the long-term implications are. So we're testing that right now on our babies. Great idea. But we'll see how that goes. On the other hand, we have people that say, well, you know, this whole human milk composition stuff, do we really need to study this? This is really important. Should we put our focus somewhere else right now? Do we need to know what's in human milk? And I give you an example here from a study that we published just recently in the last year, where we were surprised to find that there is pathogens out there. This is from a study that was done in India on rotaviruses. There is a rotavirus strain out there that starts to take advantage of oligosaccharides in human milk. And we only found that by looking at the composition of human milk. It's specific oligosaccharides that increase the infectivity of this rotavirus strain. So only by knowing that that's happening, we will be able to come up with strategy to fight that pathogen. Because otherwise that pathogen is going to win. So I think it's very important to know what is a human milk and how this all works. So basic science is important because it has public and global health relevance. All right, what are the biggest challenges facing our field? I think everyone brought this up. Funding. Again, apparently oligosaccharides well funded.
Compared to everything else.
Compared to everything else, that's true. So I did my postdoc in congenital disorders of glycosylation. Hudson Freeze, Bernadette. There's a few cases that kids have inherited disorders of glycosylation. You have 2 or 3 cases worldwide. So talking about no funding for this kind of research. The funding we have right now in the field is wonderful compared to that. But still, there is great limitations. Nathan mentioned that earlier. The TED program, for example, we started to invest $1 million in the program. It's underutilized. So there's funding put in the field, but it's not utilized very well. So I think we need to think about that a little bit, how we can use sources that might be available a little bit better. I think acceptance is very important as well. I got this, and we saw this, I think Amy showed that in her presentation, that reviewers would say, like, hey, what are you studying there? I got that from the NIH as well. It's not that important. What do you think about breastfeeding? Who cares about this stuff? And it was literally written there. And I mentioned that earlier. I think by reaching out to other people, to other fields, and dragging them into the field and showing them how cool it is, speaking at their conferences, not just at ours, will raise that acceptance. Publishing a paper, maybe not in the typical Journal of Human Lactation American Breastfeeding Association, but publishing a paper outside of our field, and ideally in a high-impact journal, that will raise its acceptance over time as well. So the only word I have there is persistence, and just keep going. And my summary would be, I see this very positive. I think this is a time of really unprecedented opportunities that we have to advance the field. from hearing all the different people in the field, yes, there's still struggles, of course, but I think we are at a spot where we haven't been before. I'm not that old to say that we've never been there before, but I think this is a great time to be in this field and to bring a workshop like this where we're trying to connect and advance the field together. I think that's beautiful. Thank you.
Okay, so I have a question for all three of you together, actually, because I think there's so many things that you can work together, and I'm hoping to work with you on this as well. So starting from what you were presenting last about the fact that with human milk composition and oligosaccharides, you're really trying to answer both questions of what drives in terms of maternal exposure and things you could modify potentially, but especially if you found some of those which are more key for infant and maternal health later on. So both sides of the equation. And in order to do that in an unconfounded way, you're going to need to find, I think, as well as doing observational research, either take advantage of trials in some way, or, and at the same time, use the genetic anchors for your human components. So both of Shelley and Meghan have talked about their GWAS of human components, and I was wondering whether you already started to think about placing it all together in that chain for mediators from maternal exposures down to fruit, which components, down to offspring effects later on.
Yeah, so we've done that actually with both of them. That's the cool stuff. So we do have genetic data for both of those cohorts, Inspire and Child. And so we're trying to puzzle this together by looking at two different cohorts. We're doing transcriptomics as well, combining that with genomics, and then doing a systems biology modeling approach to puzzle this all together, just to see how the synthetic pathway comes along, what genes are involved in that. You'll be surprised, there's an entire biosynthetic pathway that hasn't been discovered yet. So we know everything about glycosylation from all other tissues. We don't know how human milk oligosaccharides are assembled in the mammary gland during lactation. So there's a page missing in the biochemistry textbook, and not surprisingly, it's not lactation. Yeah, we're working on that.
I think once we discover these genetic markers of influencing composition, you're right, then we can apply that to other cohorts that don't necessarily have the milk data, but now we know important things about the genetics that are contributing. Also, I think it came up in a comment at some point, but this idea that components in milk might influence lactation success. So we've found, for example, in Child, one particular oligosaccharide where its level is associated with the total duration of breastfeeding, and so which way the causal arrows are pointing, like, not sure, or if that's a marker of mastitis or something else that's influencing duration, but we just recently saw that in another separate cohort, finding the same oligo also associated with duration, so these are all kind of interesting.
So I have a question for Shelley. Shelley, can you tell us a little bit more about what was the purpose of your anthropological component in your study, and can you share any general findings from that work?
So our main idea was that we assumed that environmental microbial exposures would influence the milk microbiome. So that was sort of the general idea, and that those microbial exposures would be due to both interactions with humans and interactions with animals and the general environment. So we basically tried to ask as many questions as we could related to interactions with people and animals and things like that. And we had some data from the Central African Republic, which just got published, showing that the greater the allo-maternal network, so the more people that took care of that baby, was associated with an increased microbial diversity in the milk of the baby, in the diet. So the idea there is that the baby's probably coming in contact with more microbes because there's more people holding him or her, et cetera. And those microbes are then getting transferred into the mammary gland. I mean, that's sort of the theory right now. So that was the basic reason that we set up those questionnaires. And we have loads of data that we're just barely starting to look at.
Too much data.
We have too much data. Yeah, seriously, if you're big data people and you have some questions that you can answer with our data, especially in Africa. So in Africa, for example, in Ethiopia, we have a rural setting where there's no electricity basically, and an urban setting, you know, compared to the US, they're both rural,
Right.
but very different sort of living situations. We have the same thing in the Gambia, so that's the MRC sites. And then it was a mix after that. Peru was outside of Lima. So it's a mix. So we have a lot of data. You know, we do have a large cohort that, you know, had animals in the home, certainly on the property.
And do you
No
see
running
any
water.
relationship with latitude?
Oh gosh, we haven't looked at that at all.
And changing a little bit the anthropological angle of the question, were there any societies that refused to give milk, or did you encounter any kind of beliefs that may preclude?
No,
this
but
type
they
of-
were self-selected, right?
Yeah.
So this was, I reached out to Andrew and Gloria and people that I knew were already collecting milk. So
Okay.
we didn't try to go anywhere difficult like that.
On the latitude question, it reminded me that, so at a conference, I saw a group doing research in the Gambia, and they showed that HMO profiles there were different in wet and dry season.
Oh, interesting.
So I thought, oh, I should look at season. Certainly very different in Canada. We don't have wet and dry season,
Yeah.
but we do have seasons. And
Yeah.
Very cold season.
but in the interpretation of their data, finding differences was that, oh, the women are losing and gaining so much weight in these two very different seasons.
Yeah.
We don't have that happening in Canada, but we did find one HMO that has this beautiful cycle through the seasons. So we
Wow.
don't know what that means, but it's just this power of networking and trying to ask different questions based on it.
Wow.
And you know, we found in previous studies that vitamin D, maternal vitamin D intake
Yeah.
appears to be related to milk microbiome.
So you should see a latitude difference.
Yeah.
Yeah.
I don't know. I mean, that's one hypothesis that could be checked.
Did you have any twins?